The currently approved AAV vector gene therapies for hemophilia target hepatocytes. They deliver transgenes encoding coagulation factor VIII or factor IX to hepatocytes to establish endogenous expression of those proteins, which are missing in people with hemophilia. This gives rise to two major clinical concerns. Number one, how to optimize long-term transgene expression, and number two, how to minimize the risks of gene therapy to liver health. Importantly, most adult people with hemophilia have pre-existing liver disease because of chronic viral hepatitis or history thereof, contracted from factor replacement therapy back in the 80s when it wasn't virally safe, or because of metabolic dysfunction associated steatotic liver disease, formerly known as non-alcoholic fatty liver disease. Now, people with significant liver dysfunction, such as fibrosis score 3 or 4, were excluded from clinical trials, but moderate liver disease seems to be permissive for successful transduction and transgene expression. But in general, we don't exactly know the impact of pre-existing liver disease and severity on gene therapy outcomes. On the other hand, we don't fully understand the impact of gene therapy on liver health either. Many individuals, especially those with hemophilia A, have liver enzyme elevations in the first year and sometimes later years after dosing, which may signify liver toxicity. But typically, these are asymptomatic, mild and transient. Liver biopsies from a fraction of vector recipients several years after vector dosing did not show any gene therapy-related abnormalities. There was no dysplasia, nor architectural distortion, or fibrosis, or chronic inflammation. But we still don't understand why those ALT elevations occur and how to best manage them, especially in hemophilia A. And that is because we understand very little about the mechanism of AAV gene transfer in humans in the first place. This is still an area where we need to catch up, really. In terms of liver health and its impact on gene therapy outcomes, before and after vector dosing, the current concepts are based on preclinical research primarily, from which we know that a high turnover of hepatocytes, which are otherwise rather quiescent cells, may be detrimental to long-term transgene expression. For example, in mouse models of liver injury after gene therapy, most transgenes are lost during hepatocyte cell division, which results in reduced expression of the therapeutic gene. So our understanding is that accelerated hepatocyte turnover associated with ongoing liver disease could potentially result in the reduction or elimination of the transduced hepatocytes, and therefore loss of the therapeutic effect. And this is really why optimizing and monitoring liver health is reasonable in the context of gene therapy. Now, how is this accomplished? There are no consensus recommendations yet, but the current expert opinions suggest that gene therapy candidates or vector recipients will avoid alcohol and hepatotoxic drugs. Current strategies to evaluate liver health include measuring liver aminotransferase levels, using calculations based on standard lab values such as AST to platelet ratio, using specialized proprietary liver fibrosis tests such as FibroSure or HepaScore, and liver elastography using either ultrasound or MRI. All these liver health evaluations should be performed in collaboration with a hepatologist. Avoiding acute insults to the liver before vector dosing is not only important in terms of maximizing chances for favorable outcomes, but also for accurate determination of baseline values before vector dosing. This will help monitor liver health after dosing, especially in individuals with pre-existing liver disease, which is prevalent, as I mentioned before. After dosing, vector recipients should additionally have their factor levels monitored, since a sudden decline in transgene expression may be a sign of hepatocyte death. And periodically, every 6 months, they should be screened for hepatocellular carcinoma. There is no evidence currently that AAV vectors can trigger cancer in humans, and most administered vector particles establish expression as extrachromosomal episomes, but some vectors do integrate in the genome, which raises theoretical questions about genotoxicity and oncogenicity. So screening for hepatocellular carcinoma is warranted.